Good morning. I'm Katie Angarone. I serve as the department's Associate Commissioner for Science and Policy, and I oversee the DEP's Division of Science and Research, and I also lead the Task Force on Harmful Algal Blooms. I want to thank you all for joining us today. I am delighted at how many people we have in the room. I think everyone here shares the same goal, and it's the goal that we focus on every day at DEP, which is protecting the public health and the environment. And this is why our time together today, I think, will be so well spent. We will be sharing information, replicating successes, and addressing challenges. And together, we can make sure that we reduce the impact that harmful algal blooms have on our way of life, from helping people to make better decisions about how and when to get in the water, to using the best available science to reduce future blooms. I'm confident that the information we share here today will help New Jersey keep its residents and visitors safer. As you're aware, harmful algal blooms have impacted lakes and beaches globally and nationally, as well as locally here in New Jersey. These harmful algal blooms are the result of excess growth of cyanobacteria, usually caused by increased nutrients, sunlight, and water temperatures. And climate change, we know, can create the conditions that are favorable for the growth of HABs. In New Jersey, not only are we seeing an increase in the occurrence of HABs in our freshwater, but we're also seeing an increase in HAB persistence, with many of these blooms lasting into the fall and some of them even overwintering. And research suggests that the impacts of climate change may promote the growth and dominance of harmful algal blooms through a variety of mechanisms, including warmer temperatures, increases in atmospheric carbon dioxide concentrations, and changes in rainfall patterns. Additionally, we know that HABs are not only an environmental issue. Their presence severely impacts not only our public health, but our economy. So DEP has made significant progress in addressing HAB occurrence and response in New Jersey. While 2020 saw another upturn in HABs, including an increase of HABs on drinking water sources, DEP has done a better job communicating to lakes managers, mayors, and the public about the health risks associated with HABs and what people can do to minimize risks to themselves and their pets when recreating on New Jersey's waters. DEP provided $6 million in grants through 2 RFPs in 2019, and much of that funding supported innovative HAB strategies, some of which were implemented during last summer. The results of these projects will be shared today. It's important to remember that there is no single answer in dealing with the over-enrichment, climate change, and HABs, and it will take a holistic watershed approach to manage the problem. A core principle at DEP is to base our decisions on the best available science, and this applies to HABs as well. DEP has updated our toxin thresholds based on revised exposure criteria developed by EPA. These new toxin levels, and in addition with a new threshold for saxitoxin, will be presented today and will provide for protection for the public's health and welfare. DEP has put together an independent team of HAB experts to assist in ensuring the best available science at its disposal. Additionally, we have undertaken a targeted reorganization to bring certain elements of our land use and water resource management programs into closer alignment under a reimagined watershed and land use management program. Our vision for this program is that through a holistic approach to planning, permitting, mitigation, and restoration of impacts in New Jersey's watersheds, we will work to preserve, protect, and improve the integrity of New Jersey's water and natural resources. That includes taking a holistic approach to reducing the impacts of HABs in our water bodies. The goal for this year's summit, and I'm delighted to have a second one, is to focus on how to address HABs by looking at successful lake management strategies, identifying helpful best management practices, and showcasing the most emerging science and technology available. All of this is meant to help you and your communities to build a partnership that will allow us to prevent and respond to HABs in the future. The speakers that you'll hear from today will go into more details on many of these topics, including our keynote speaker, Dr. Jennifer Graham from USGS, who will touch on the impacts of climate change on HABs, updates from DEP staff, mitigation measures and success stories, impacts of HABs on drinking water, funding, and much, much more. And we'll have a Q&A period at the end of each session, and we look forward to hearing from you. That is so valuable for us. So today's summit is really just one more step in a continuing conversation about HABs in New Jersey, and I truly look forward to working with you and learning from you. So with that being said, I will hand the mic off to Carrie Kirkflew, who will act as our MC. Thank you, Carrie, today. And she will go over some logistics and how to use the platform to make this summit as interactive as possible. Thank you so much for attending.
Good morning, everyone. It's nice to be here on this beautiful sunny day. Happy to report that we have a full house. Fully 250 people have joined us, so this is pretty exciting for us. And we're going to hopefully have a productive discussion within the confines of the platform that we're using. With that, I just wanted to say that we are using Microsoft Teams if you are unfamiliar with that. The way that we would like to have you be involved is actually twofold. If you look at the top of your screen, you'll see a little hand, and you'll also see a little box, a chat box. If you wish to ask a question, we ask that you click on your hand, and at the conclusion of each one of the sessions, we will call upon you. The other way to be involved is to write your question in the chat, and we have someone, myself and a couple of other people, taking a look at the chat, and we will relay your question to the speaker at the end of the sessions. I'd like to remind you that we are recording the session today. This is to benefit those who are unable to come, and also as a resource for you later on if you need to go back and take a listen to one of the presentations that we're recording today. The other thing to keep in mind is that this is a full day program. We will take a break at lunch, and then we'll be back, and we ask that if you do leave the session that you click on the same link that you did this morning to join us. And we hope that you'll be able to join us the entire day, but we certainly understand if you're only able to stay with us this morning. And again, I remind you that we are being recorded. So with that, I'd like to turn it over to our keynote speaker, Dr. Jennifer Graham. Dr. Graham currently serves as the Harmful Algal Bloom Coordinator for USGS Water Mission Area. She also represents USGS on the Interagency Working Group on Harmful Algal Blooms and Hypoxia. And with that, I'd like to turn it over to Dr. Graham.
Good morning, everyone. I'd like to thank New Jersey Department of Environmental Protection for inviting me to be part of the Harmful Algal Bloom Summit today. I've worked in harmful algal blooms for over 20 years, and I've been in a unique position over the course of my career to watch the science evolve and see how our conversations and discussion about harmful algal blooms has changed over time. As already been mentioned this morning, harmful algal blooms are a global concern and can affect a range of aquatic ecosystem types from headwater streams to coastal waterways. Harmful algal blooms are complex and present many challenges, not the least of which is that they are formed by unicellular organisms, which can accumulate until they are visible by the naked eye and even from space. So there are many processes occurring across a range of scales. There are many different types of algae that can form harmful blooms, and I've spent my career focused on the Cyanobacteria, which are often what we think of when we think of harmful algal blooms in fresh water. Next slide. So cyanobacteria were first noted to produce toxins after sheep deaths in Australia in 1878. Shortly thereafter, cyanobacteria-related animal intoxications were reported from the United States, and throughout the 1990s, reports occurred every so often from around the country. I'd like to highlight some of the work that was done in Iowa lakes in the 1940s and 1950s when severe blooms were observed to the point that there was a recreational beach monitoring program in Dickinson County, and in-lake management actions were taken to control blooms. The study of these events in Iowa in the 40s and 50s contributed to some of our foundational understanding of harmful cyanobacterial blooms in the United States. These key findings included the recognition that cyanobacterial blooms are a nuisance issue that can sometimes be toxic, that these events are usually, but not always, a summer phenomenon, that high nutrients and other ideal ecological conditions result in blooms, and even the identification of potential producers of cyanobacterial toxins, many of which we still talk about today, like aphanizomenon, anabina, and microcystis. One of the things that really stands out from this time is that while there was recognition that the toxins produced by cyanobacteria were complex and that there were many different types, they concluded that it was almost impossible to determine what they were at that time. So they knew they were dealing with the first group of toxins, but they did not know what they were. As such, the recreational beach monitoring programs would rely on mouse tests, and they would collect a sample of cyanobacteria, inject a mouse, and if the mouse died, people did not swim that day. Now, there was a lot of work done in the 40s and 50s, and then the recreational beach monitoring program petered out in Dickinson County. I don't know the history of that, and it wasn't until the early 2000s that recreational monitoring was picked up again in Iowa. And management actions, again, a lot of activities in the 40s and 50s, and then that picked up again in the early 2000s. So I want to contrast what we knew in the 40s and 50s, that this was just an occasional phenomenon, something that sometimes happened, as well as what we knew, when I entered into this field. This is something that happened from several states throughout the United States, but it was not a ubiquitous phenomenon. And today, harmful blooms and cyanotoxins have been reported from all 50 states in the US. Next slide. And harmful algal blooms are considered a serious water quality threat to public and aquatic ecosystem health globally. Harmful algal blooms have increased worldwide. And although this phenomenon has been observed, certainly anecdotally, it's been difficult oftentimes to quantify this increase because awareness, reporting, and measurement of blooms has also increased. Next slide. However, there are data documenting the increase of blooms worldwide. For example, this is a study by Jeff Ho and others that used satellite imagery to quantify the change in peak summertime bloom intensity and found increases in many lakes worldwide, as indicated by the pink circles and red triangles. And this has been looking at change since the 1980s. Next slide. And the state of Kansas has collected cyanobacterial abundance data for over 30 years, and all of their data has been identified and counted by the same analyst nonetheless, which makes it a really unique data set. And in a retrospective analysis based on Kansas Department of Public Health advisory and warning criteria, the number of health alerts that would have been issued in 1989 and 1995, between that time period, was 13. And note that that number increased to 25 between the time period of 2003 and 2009. So there's some clear data on the increase of cyanobacterial bloom events in the state of Kansas. They have a nice data set to be able to document and quantify that. Next slide, please. One of the questions that we often ask is since cyanobacterial abundance and blooms are increasing, has cyanotoxin occurrence and concentration also increased. And documenting increases in cyanotoxin occurrence has been more challenging since we really haven't been measuring the cyanotoxins for a very long period of time. And often we collect cyanotoxin measurements in response to events rather than as part of routine monitoring programs. However, this is an example from the University of Missouri Statewide Lakes Assessment Program that measured microcystins during 2004 and 2006, and again during 2017 and 2018, using the same basic study design. Between 2004 and 2006, about 25% of all samples collected had detectable microcystins, compared to 60 to 80% in 2017 and 2018, suggesting an increasing in occurrence. I do want to contrast that with cylindrospermopsin, which was measured in 2006 and detected in 14% of samples, which was similar to what was observed in 2017 for cylindrospermopsin, but much less than what was observed in 2018. And this example just highlights the temporal variability in cyanotoxin and harmful algal bloom occurrence, and how much that variability can affect our interpretation and understanding of what's going on. Next slide, please. As mentioned, cyanobacterial blooms are increasing and are anticipated to increase under various scenarios of climate change. And this is just one example of a study led by Steve Chopra that found that cyanobacterial abundance under all scenarios of climate change they tested were anticipated to increase substantially by the year 2090. The models used to protect change in this study included a range of water quality variables known to be important to cyanobacterial bloom formation, and included scenarios with substantial reduction in greenhouse gas emissions and scenarios with substantial increases in greenhouse gas emissions. And so if you look at the figure here, the black line is just control conditions. So that's a scenario of no change. The green lines are a scenario with reduced reduction in greenhouse gas emission, and then the yellow lines are increased in greenhouse gas emissions. And you can see that under all scenarios of change, summertime cyanobacterial abundance is anticipated to increase quite substantially. Next slide, please. And so, as was mentioned in the opening remarks, we broadly know the environmental factors that are associated with cyanobacterial bloom formation, and they include nutrient loading to our aquatic ecosystems and how those nutrients are cycled and processed. Temperatures are important. Water column physics are important, light is important, and food web interactions are all important. And we know these things broadly, but it still really remains a challenge to be able to identify exactly when and where a cyanobacterial bloom will form, how long that bloom might last, and whether or not that bloom will produce toxins. And climate change scenarios add all the more to this complexity. We know that climate change will result in increased temperatures. These increased temperatures will lead to more intense and longer periods of stratification in lakes. And we know that the timing and magnitude of nutrient delivery to systems will also change. So there may be more intense storms and longer periods of drought. And so we'll have intense period of nutrient loading followed by periods of drought. And all of these changing conditions may favor the cyanobacteria. Added on top of that, our paradigms are changing in the way we think about nutrients and environmental influences on our aquatic ecosystems as a whole. And this includes harmful algal bloom formation in both our freshwater and our marine systems. And just as a couple of examples, We now know that the types of nitrogen and phosphorus present in our ecosystems are important, not just total concentrations. And we know that when these nutrients are delivered to the system is important. And we're starting to see cyanobacterial blooms happen in nutrient-poor systems. These are places where 20 years ago, I would have told you cyanobacterial blooms were not a concern, and we're seeing them in these systems now. We're also seeing freshwater cyanobacteria and cyanotoxins transported hundreds of miles and affecting marine systems. So our coastal and marine environments are now experiencing not only the unique harmful algal blooms that occur in the marine environment, but also the transport and importation of freshwater toxins. So there's a co-stressor in marine systems from freshwater and marine influences on HABs. And all of these changes are influenced by climate change. And so in thinking about influences and thinking about how we design our studies and how we mitigate and manage, we always need to keep in mind the possibility for change and how a changing climate may influence bloom formation. And I wanted to spend a little bit of time talking about the cyanobacteria and some of the adaptations that they have. Cyanobacteria are ancient organisms. They're billions of years old. They're diverse. There are many different species and strains of cyanobacteria, and they're highly adaptable. As a group, the cyanobacteria have many different traits that allow that favor their occurrence and dominance in many different kinds of situations. These traits include a temperature tolerance. They can tolerate higher temperatures than many other algae. They have buoyancy control, so they can change their location in the water column. They can store phosphorus. They can fix atmospheric nitrogen. So next slide, please. I'm sorry. And slide.
Just letting you know you have 5 minutes, okay?
Okay, slide after that, please. So they have access to nutrients that other organisms don't have. They can produce resting stages, can capture light at low levels, resist grazing, and can produce toxins. And so many of these adaptations allow cyanobacteria to be well suited to changing climate conditions. And I'll touch on just a few examples. Next slide, please. So temperature controls all algal growth rates, and increased temperatures lead to increased growth rates to a maximum tolerance level. Cyanobacteria achieve maximum growth rates at warmer temperatures than other types of algae, and even at temperatures where other algae really don't fare well. So on the figure here on the right, cyanobacteria are on the bottom, and a couple of different algal groups are on top of that. Next slide, please. Cyanobacteria also have many different ways that allow them to control their location in the water column, allowing them to take advantage of optimal conditions for growth, and temperature influences water column structure. So longer and more intense periods of water column stratification will favor buoyancy regulating cyanobacteria. Next slide, please. Cyanobacteria are also known as ecosystem engineers. They have several adaptations that allow them to access or store nutrients. And as such, they can bring nutrients from the atmosphere or the bottom waters into the water column. And other algae don't have these adaptations. These adaptations have led to the description of cyanobacteria as ecosystem engineers. So the cyanobacteria themselves are driving some of the changes that we are seeing in bloom occurrence. Next slide, please. We're really just starting to understand the independent and interactive influences environmental conditions have on harmful algal bloom development and how changing climate conditions will affect these combined influences. Just as one example, we know that warming temperatures will favor the cyanobacteria, but some studies show that cyanotoxin production actually decreases with increased temperatures. But layer on top of that the fact that the sensitivity of aquatic organisms to cyanotoxins may increase with increasing temperatures. So it's complicated, and there's still a lot of research needed to fully understand cyanobacterial blooms and their impacts, let alone how things may change as the climate changes. Next slide, please. So I want to come back to some of the work that was done in the 50s. And one of the conclusions from that work was that a considerable amount of research is being conducted toward the determination of causes and control of algal blooms. And the expansion and integration of these studies is indicated as a very real need. And that statement is as true today as it was in the 50s, even though our knowledge and understanding of cyanobacteria, the cyanotoxins, and their influences has so greatly grown since this time. There's still a lot of research needs. There's still a lot we don't know about status and trends, environmental fate and transport, environmental drivers, ecosystem effects, exposure and health, drinking water and food impacts, mitigation and management, and overarching all of this, how the interactive influences of climate change may influence all of these things. So again, cyanobacteria are unicellular organisms that can accumulate to the point where we can see them with the naked eye and even from space. And the factors influencing this accumulation are diverse and complex. Next slide, please. And so, as such, New Jersey's put together an expert team to help better understand and manage harmful algal blooms. And this team will be introduced to you later in the workshop today. Next slide. With that, I'll take any questions if we have time.
Dr. Graham, I think we're going to hold questions until after Vic, and then we'll have questions for both you and for Vic. Okay. So folks, just hang on to your questions or put them in the chat, and we'll call on you after Vic Peretti speaks. And with that, Vic is our section chief for the New Jersey Department of Environmental Protection's Bureau of Freshwater and Biological Monitoring, where he oversees the sampling and analysis of freshwater monitoring throughout the state. Vic?
Thanks, Carrie. Today I'm going to give a brief update on our enhancements and progress from 2020, which will continue into 2021. Next slide, please. So in 2020, we developed a tiered public information and signage system as an enhancement to our HAB strategy. The alert tiers provide clear guidance on suitable recreational activities and impact to water bodies, depending on the levels of cyanobacteria and/or cyanotoxins present. The color-coded signs provide public with current conditions and recommendations on which recreational activities are suitable and those that are not. The index makes it clear to the public that in some instances, such things as boating and related activities may still be suitable when lower levels of harmful algal blooms are detected. Next slide, please. We developed a winter watch sign this year for the off-season. This is intended to be used at water bodies where HABs are still persisting or have a likelihood of reoccurring during the winter. The sign cautions users of conditions that may be present but not yet reported or confirmed. We also felt this new sign provided a fresh perspective during the off-season, and it would be obvious that the sign was not old or left over, so to speak, from the summer season. It's available to partners and communities who feel it's necessary at their water bodies It can be downloaded, or you can request a plastic sign directly from us. Next slide, please. The HAB interactive mapping and communication system is used to report suspected HABs and display data and alerts. The new reporting system and map have been extremely successful in 2020 and very widely used. It's used for reporting a suspected HAB. It can be used with a smartphone or PC. Areas of concern can be exactly pinpointed. It provides photos, examples of HABs to compare what you're seeing in the field. Photos that you take on site could be uploaded into this report. There's a line that could be submitted for observations and field data, and it provides a lot more information to us than just reporting it to the hotline by phone. Reporting it through this system, an immediate email goes to DEP HAB staff. We have a faster, more precise response and a more efficient use of our staff resources. Next slide, please. The data and alerts are uploaded to the dashboard as soon as the results are available. There's a tutorial link on the dashboard site to assist you with its use. For 2021, the 2020 map will be archived. It'll still be accessible on the website with most of its searchable features remaining. A big enhancement for 2021, there's a download function. It's for current and past data. It's available right now. This is what it looks like. There's a link on the active live dashboard on the bottom left-hand corner. You follow the link for data downloads. It'll take you to this web page, and you can download site by site. You could do download of everything we have. So depending on what you're looking for, all of our data is now available through this website. Next slide, please. We're expanding our continuous monitoring program. Continuous buoys provide real-time water quality data for HABs using phycocyanin pigment measurements. These buoys also provide water quality conditions such as pH, dissolved oxygen, temperature, conductivity. To provide context to the phycocyanin readings, we developed correlations between phycocyanin and cell count approximations. I just want to make note that each brand of meter will have a different correlation developed. Each model has different ranges and precision. So a YSI continuous meter, which we use on the buoys, would have a different correlation than, let's say, a Turner Designs field meter. So this chart, cells per meter, cells per mil, comparing it to the meter readings, is for the YSI continuous monitor. And here you see an example of what you can download from the website. This is Manasquan Reservoir. You could download any or all of the water quality parameters, including the phycocyanin, and you can download into a graph such as the example here. In 2020, we deployed buoys at Manasquan Reservoir. Capaccon, for a period of time, it was unfortunately damaged. There was one at Spruce Run Reservoir that was provided by USGS. In 2021, we anticipate 10 more buoys being deployed throughout the state. And this once they're deployed, again, this information will be available live via our continuous monitoring website. Next slide, please. Each year, the most current science is reviewed. Enhancements based on this review will be incorporated into the 2021 strategy, which is anticipated to be released in the spring. Although there were maybe slight changes to the toxin thresholds, there will not be significant changes to the alert tier system. There's a science session later in the summit and a presentation on cyanotoxins, and they will also be discussing the derivation of the new toxin thresholds. Next slide, please. To communicate our strategy, we continue to develop and enhance our outreach. Outreach materials were printed and provided to some of our partners, including parks, Division of Fish and Wildlife, and local health departments. They are downloadable and available upon request. If you need actual posters or alert tier signs, we could provide those. We created 3 training videos, and they are also available online. There's one on how to report a HAB using our HAB reporting system, the use of the phycocyanometer, and how to take an actual HAB sample for laboratory analysis. We also performed 3 training workshops. We performed one for parks and DEP, Parks, Division of Fish and Wildlife, and Compliance Enforcement. Some of our outside partners, Department of Health and local health departments, and we also had a training workshop for citizen science volunteers. And we anticipate that these workshops will continue in the future. And all this information on our outreach, our alert tiers, can all be found on our new website. And Elena Baldwin-Brown will present next and show you all of our enhancements to our website. Thank you.
So Vic, with that, thank you, Vic, very much. We're going to pause for a minute, and we're going to take some questions. If you have a question for Dr. Graham, this would be your time to ask a question, both of her and of Vic. So I see in the chat, Leslie, you had a question. Do you want me to read it, or would you like to take your mic off and ask the question yourself?
Hi, Carrie. I can ask the question.
Go ahead and ask your question.
Thank you. Hi, Jennifer. Great presentation. You described challenges in quantifying temporal trends over time for blooms and toxins, as we haven't been measuring them in the same manner year after year in many cases, or toxins not until more recently. I just wondered if there are national or international surveillance systems now set up to systematically measure bloom or toxin occurrence or the duration, and if so, what are the metrics that are being used, or if there isn't anything quite set up like that yet, what would be the best metrics to use to look at long-term temporal trends?
Yeah. Hey, Leslie. So the short answer to your question is no, we do not have national or international surveillance systems yet that are routinely reporting a universal metric. That is something that I have heard come up in discussions, but trying to determine what that metric is a challenge, because there are different types of blooms with different endpoints. So you could measure abundance of an organism of interest, you could measure overall algal biomass, or you could measure one of the toxins. So, you know, there's discussions of what should we measure, what should we always measure, and there really is not an easy answer to that question. And I don't know that I could even make a recommendation, because it you know, to a certain extent, it depends on what the organisms of concern are in a given area.
You did mention something about a satellite study on intensity, and I just wondered if that had any promise for something like this.
You know, satellites do have promise for status and trend work because they are consistent over time. There are, as with anything, you know, there's challenges and opportunities there. The study that I referenced looked at overall algal biomass using chlorophyll as a surrogate. And so that is one indicator that we can go at least back into the 80s and look at change over time that way. To look at different types of blooms, for example, to look at cyanobacteria blooms or some of the blooms that are occurring in the oceans, we don't have as long of a time period for those satellites, so that the reference time period there is shorter. But there's also promise there. So again, you know, there's challenges. They aren't, there's some error associated with that, but they are consistent and would give a global indicator.
Thank you, Dr. Graham. Vic, we have a couple of questions for you. We have one from Fred Lubnow. Fred, would you like to unmute yourself and ask Vic the question, or would you prefer my reading it?
Either way, I mean, can people hear me?
We can all hear you. Go ahead.
Okay, yeah, and I think Vic actually may have already answered it in the chat, but I was asking, I noticed the 2021 alert table had the alert level removed compared to the 2020 table. But Vic, you mentioned that the alert is still there, but it's for internal purposes, which initiates additional monitoring at bathing beaches. I didn't know if you wanted to elaborate a little on that.
Sure, I can. So the alert it's in our strategy, and it can be viewed in our strategy if people want to look at it. It's intended to alert us to, so it's specifically for bathing beaches. If a cell count starts approaching over 40,000 and it's getting to the 80,000 range where a beach will be closed, it alerts us to do additional monitoring to keep an eye on it, just so that most appropriate alert is in place, and if a beach closing has to take place, we're on top of it. So it's not an actual posting that we post, it's more or less the watch posting, but for beaches it just gives us a little initiates additional monitoring.
Great, thank you.
Vic, we have another question from Christina Reeves. Christina, do you want to share your question?
Vic answered it in the chat there. He put the link
Okay.
in. I didn't know if you needed the actual app, the Survey 123, but now that I'm on my smartphone looking at it, it just goes right to it as part of the web browser. So I see that. Thank you, Vic.
You're welcome.
Okay. Brad, you have a question? How do people differentiate between algal bloom and HAB visually? Is there anything further you would like to add to your question? You can unmute.
Well, actually, I had two questions.
Okay,
The first, can you hear me okay?
Yes.
Great. I had two questions. The first one had to do with the continuous monitoring device. And I'm wondering if that's available for private lakes, since we're told that we own the land under the water, but the state owns the water. Hence, we pay things like fees for registering boats and things like that. So the first question is the monitoring device available for private lakes? The second one is, our members are concerned about HABs, but we also just have regular algae blooms. And so I'm wondering, how does the layperson differentiate visually from a what I call a regular algae bloom versus an HAB?
So for your first question, like I said, we're going to have 12 total buoys available for deployment this year. They're going to be at priority lakes, which have reoccurring HABs, have high levels of recreation, drinking water sources, things like that. So they'll be deployed where the most where we get the most reoccurrence and the most risk to users and drinking water users. For your second question about how do you determine visually what a harmful algal bloom is, on our website we have some examples of what harmful algal bloom looks like and what it doesn't. Some common things like duckweed that might be mistaken for a harmful algal bloom. So you can check that out. Usually it looks like spilled paint, it has a blue-green appearance, neon green. One of the good tests is called a stick test. Usually regular algae has a form and a mass. Cyanobacteria doesn't. If you put a stick in it and you're able to pick it up, it's usually not a cyanobacteria. It's usually like filamentous algae or green algae. Later in the program today, we're going to have a phycocyanometer loan program. It's a field meter, handheld, very easy to use. You can dip it in the water and get a reading, and you can tell whether there's a cyanobacteria bloom present, or at least estimate it. So that's going to be another program available to our partners to help them determine if there's a bloom at their water body.
Thank you very much.
Okay, I think we're going to stop there and continue on with our discussions. But before we do, I'd like to thank Dr. Graham for her incredible presentation, her keynote address. Thank you, Jennifer. It was very informative. And thank you, Vic, for the updates from DEP so far. And we're going to move on to some more with Elena Baldwin-Brown. Elena currently serves as our communications manager for the DEP Division of Water Monitoring and Standards, and she's going to be giving you an update on our website. Elena?
Thanks, Carrie. Good morning, everybody. My pleasure to be with you here today to walk you through some of the changes that have been occurring over the past year with our website. So I'm hoping everyone can see my screen right now, so what I wanted to do is, many of you may already be familiar with and using our HAB's website fairly frequently, but for those who are not, I just wanted to let you know it's very, very easy to access. If you are on the main DEP homepage, which is what I'm showing you right now, if you scroll down to the bottom on the right-hand side, you'll see a link that says harmful algal blooms. And if you click that, that will take you right to the main DEP HAB's webpage. So for those who have used it or have seen it in the past, it may look a little different because this fall we gave it another facelift. We did some enhancements, some sprucing up, some reorganization of material that had been there, some repackaging of things, just to make it a little bit easier for folks to get to things that were probably most important, as well as grouping things so that users didn't have to search all over the place and it might be a little bit more intuitive for them. So let me first share with you what we added to the web page, and then I will let you know what has become more prominent. So the first thing that we added, and this was as a result of some feedback that we got from those of you who attended the summits that we held in 2020, was you asked for a HAB listserv. You said you were interested in finding out news and information about what was going on in DEP. So what we've done is we have created one, and there is a very easy link to sign up for it right here on the left side of the web page. We also heard from you that people were interested in learning what they can, or their constituents, or their kids, or their families, or their organizations can do with regards to HABs. So we added a button right here at the top that when you click it, pops open a button, gives you some ideas and some links to different things that you can avail yourself of. Again, including the listserv that I just talked about, the report a HAB that you heard Vic talk about, and the photos that were just discussed prior to my starting this presentation. So we added that to the website. Hopefully makes things a little bit easier for folks to get to. You heard Vic Peretti talk about the training videos that we put together this summer and are now available for people who, and they are right here, front and center. If you click on the more information button, it just takes you to the spot where you can find the Report a HAB training video, which is general video for the public. For those who might want to, who are interested in learning how to take phycocyanin measurements, and for those partners that are actually doing HAB sampling for laboratory analysis. And then, as you'll hear more later on today, well, Dr. Graham already alluded to it, and you'll hear more from both Rachel White as well as some other folks during the day. We now have a page that talks about our expert team that was recently developed. So what's more prominent? Again, those of you who have been using our HAB's website, what else do you have? What can you get to more quickly now? Here is the link to that, the mobile version of our interactive HAB reporting map. We heard from stakeholders and from partners that accessing it from their mobile phone wasn't perhaps as friendly as it could have been. So we've added that and made it more prominent for everyone. Probably the two biggest things are, as Vic was talking about, our HAB interactive reporting and communication system. This is where you find all the results. Many of you probably used this over the summer and know exactly what you can get, what information you can get there. But I wanted to take one quick second to show you where our new data download feature is, right here on the bottom left of your screen in the box that's there. There are a handful of different tools, but this data download feature is right here. So I just wanted to be sure everyone knew how easy it is to get to, especially from the department's HAB's webpage. Also very prominent, report a HAB. This is where we need all of your assistance. Instead of having to hunt through or look for buttons everywhere, we've made it very, very easy to get to from the department's homepage. It's also very easily accessible still from the Bureau of Freshwater and Biological Monitoring's Cyano-HABs page, but if you don't often visit that site or you've never visited that site and you're looking for a quick way to get there, here it is from the main page on the department's page. Okay, so what we've done here is try to group many of the things into categories that make sense and make it easier for you to access. The tiers and the signs that you heard Vic talk about, very, very easily accessible now right from this page. If anybody wants more information or needs to see the information again, you can get to it. You can also download in several different sizes all 6 of the signs that we have had previously available, as well as our new winter watch sign. But as Vic also said, you can reach out to himself or folks in the Bureau of Freshwater and Biological Monitoring if you'd like a plastic version of any of those signs, and they can provide them to you. We've also got our fact sheets here, and there are a couple of new ones here since probably I spoke to you last year. We've added a pet fact sheet. We've also added 2 new Stop a HAB planting guide fact sheets. These are specific to North Jersey and to South Jersey. So we have a few more resources in addition to updated general information fact sheets, as well as recreational exposure and health effects fact sheet, and the fact sheet for lakefront communities that I think any of you that attended the summits that were held in 2020, you had the opportunity to receive one of those at that point in time. Again, we have some HAB monitoring FAQs here. They're also on the newer side. There was information, all of that information was available previously, just not all in one place. Some other things that are a little bit more prominent now. You heard Katie Angarone talk to you about the grants that had been awarded, and so we have a direct link to both the grants and other funding mechanisms right here. The photos that Vic talked to you about, we've enhanced the page so that you can see what are actual cyanohabs, as well some of the organisms, and then here's what we call HAB lookalikes. May help someone who is out in the field and is looking at what they think may or may not be a HAB to determine what they've got.
Elena, we have about 1 more minute.
Not a problem. I am done. Carrie, I just wanted to say the rest of these are all, residential and municipal information. Again, it's all the same information that everyone that was there previously, just repackaged, hopefully to make things easier for users to find what they're looking for. And with that, I will stop sharing my screen and let our next speaker, let Carrie, go back to Carrie and let her continue with the day. Thank you.
Thank you, Elena. That's great. Thank you so much. So our next speaker is Rachel White, and Rachel is an environmental scientist in the Division of Water Monitoring and Standards. She is also the department's liaison to the expert team, and Rachel is going to be talking a little bit about that and introducing some of the team members. So, Rachel.
Hi, everyone. As Carrie said, I am the DEP liaison to the expert team, and the coordination of this team is done collaboratively with New Jersey Sea Grant. So my counterpart over there is Mike Danko. I'm going to give a brief overview as to why the team was established, who the members are, and what the scope of work is for the team. And then after I talk, two expert team members, Dr. Bob Cortman and Dr. Jason Adolf, will be speaking in a little more depth about specific charge questions the team is working on. Next slide. So in November 2019, Governor Murphy launched a new initiative to address the HAB problem in New Jersey. I'm sure many of you are aware. There are three components to the initiative, but the second component is to build upon the state's scientific expertise and enhance its capacity to respond to HAB events. So DEP, working with Sea Grant, recruited a team of experts from across various sectors with the goal of fulfilling this second component by evaluating and addressing prevention and mitigation strategies for HABs, developing New Jersey HABs and lakes management guidance materials, reviewing progress and outcomes of New Jersey HAB-related projects and data to identify lessons learned, and providing just general technical HAB advice, I should say general and technical, education and training to assist local partners and others with the development of HAB action plans. Next slide. So we recruited 10 members to the team from different sectors and with a range of experience and expertise that would inform lakes management and or HAB prevention, mitigation, and management. And so these team members work with an internal DEP team that has representatives from a number of our programs that work on HABs. Next slide. So the expert team's primary objective is to provide guidance to DEP on HAB prevention, mitigation, and management, as I said, for New Jersey lakes and other water bodies. And as part of that, they have a number of tasks that they are working on or will be working on. And many of the resources they will produce will be made publicly available either on the DEP website or in another format, but they will be particularly beneficial to lake managers and those who regularly deal with HABs, but they are less geared towards sort of general public use. So the first task is a literature review on the prevention and treatment of HABs, which will be formatted as a resource guide and posted to the website. They're also working with DEP staff to review DEP collected HAB and water quality data. And then based in part on the literature and data reviews, they will be developing guidance documents for HAB lake management in New Jersey. And these guidance documents will look at how to develop a lake-specific action plan, but there will not include management plans for specific lakes and their unique conditions. It's more of a general how-to for how to develop a plan. And they will also be providing technical advice throughout as requested by DEP. And then finally, they will be developing a training program for DEP staff and interested stakeholders and partners. And they will be conducting a few training workshops in different locations around the state. The format and location of those will be COVID dependent and are still TBD. So you can find a list of the team members and tasks, as Alina mentioned, as well as other information on the DEP HAB website. And I've included that link as well here on the slide. So thank you.
Rachel, you can go ahead and introduce the speakers. You're muted.
Oh, I apologize. So our first speaker is Jason Adolf, and he is one of our expert team leads from Monmouth University in the biology department and the Urban Coast Institute. Jason?
Thank you, Rachel. You can hear me? Everyone can hear me?
Yes, you're all good.
Okay, thank you. Great. So yeah, thank you for the introduction. I'm really excited to be serving on this team. We have a great diversity of backgrounds and perspectives, and I've already learned a lot just from interacting with my co-team members. Next. So I grew up in Hightstown, New Jersey, but in '89, when I finished high school, I left. And I spent my career studying HABs in marine, estuarine, and freshwater environments in a lot of places in the country and the world except New Jersey. It wasn't until I came back to New Jersey in 2017 for the job with Monmouth University, I really started looking into the local New Jersey HAB situation. And I found a couple of interesting things that I always like to share here. There was a 2013 radio interview, on the left there, and if you could, next. That pointed out that all these HABs were happening in different states, but not in New Jersey. I thought that was odd because being a HAB researcher, I knew there must be HABs in New Jersey. Then, in my local environment in Deal Lake, Asbury Park, I started to learn and work with the HABs there. And then, of course, in 2019, Lake Capaccon happened, and we quickly got into the, no one would say that there's no HABs in New Jersey anymore for sure. Next, please. So I'm essentially a phytoplankton ecologist, and what I do in my research is I look to understand how physics, chemistry, and biology interact in an ecosystem to produce different phytoplankton outcomes. And by phytoplankton outcomes, I mean what's growing in the lake, what's growing in the estuary or the ocean. HABs, HABs are one such outcome that are driven by certain sets of conditions. Next. And I also understand that because of the complexity of the problem, of course, your HAB is not my HAB, by which I mean that all HABs aren't equivalent. We call them all HABs, but Deal Lake HABs are likely very different than HABs that occur in Capaccon or Manasquan Reservoir or any reservoir. So next, there are no one solution. However, there is an overarching theme that we heard about this morning in Dr. Graham's talk that is widely accepted in the research community, that anthropogenic eutrophication, which is the loading of nutrients due to human activities in the watershed, and climate change exacerbate HABs. They make existing ones worse, and they also are responsible for the emergence of some HABs where they didn't exist before. Next. So when we're thinking about HABs and environmental causes of HABs, we have this list of ingredients: nitrogen and phosphorus, our nutrients. We have temperatures, and we have rainfall, which is implicated in both delivering nutrients to the systems as well as a factor that is impacted by climate change, changing patterns. So what I want to cover in my talk here is, what do these list of ingredients look like in our region? Bring the broad principles of climate change and eutrophication down to the state. Next. And I'll start off with the eutrophication situation in the Northeast in New Jersey, citing a study by Milstead et al., who used the National Lake Assessment data to determine the trophic status of lakes and watersheds in the Northeast, including New Jersey, which I've circled in red on these maps. And what you'll see is that, first, let's look at the trophic state scale between the two maps that run from oligotrophic to hypereutrophic. Oligotrophic and mesotrophic would be associated with lake conditions where the water is clear, oxygenated, even at night, and are unimpaired and have relatively low phytoplankton biomass, whereas eutrophic to hypertrophic would be those lakes that tend to be have impaired water quality, such as low oxygen, and tend to experience harmful algal blooms. If you look at the map for New Jersey, you'll see that a lot of our regions are coded in the eutrophic to hypereutrophic categories. Next. So in terms of eutrophication, we're starting our investigation of HABs and how to mitigate them within an environment which already favors them on a big and regional scale. And that's an important thing to keep in mind. Next. Then there are climate change indicators. Go next twice, please, Jessica. And we saw this morning. Climate change is ultimately being driven by the increase in carbon dioxide in the atmosphere, due principally to humans burning fossil fuels. And we can see that in the Mauna Loa record on the top. And on the bottom, we're reminded that relative to this 800,000-year-old record from the Vostok ice core, our present-day carbon dioxide levels are literally off the charts compared to Earth's history. Go ahead twice, please, Jessica. On a more regional to local scale, it's been observed that the temperatures in the Northeast, including New Jersey, are increasing faster than most of the planet. And the atmosphere is not a homogeneous thing. It's a heterogeneous thing, and we happen to be in a warmer, a faster warming area. And we've also observed increases in precipitation. Precipitation is predicted to increase about 10% by 2050, and that includes not just more rain, but, and importantly for this topic today, an increase in the intensity and the frequency of storms, which are the big rainfall events that really bring stuff into our lakes and our coastal waters. So any phytoplankton ecologist, lake manager, looking at these factors would have to come to the conclusion that if not addressed, the eutrophication and the climate change situation is just going to tend to make HABs worse in the future. Next. So the way we're addressing these, as Rachel described as part of the HAB and lake monitoring expert team, one of the ways we're addressing these is through data and data-driven science and decision making. And when I say data, I'm referring to agency data, such as the NJDEP data I'm going to show today, but also university partner data, and importantly, citizen science data, which I happen to think is very important in this issue, not only because it gives us more data, but also because it actively engages communities who have a different perspective on these water bodies than we scientists do. Communities who live and work there look at those lakes differently than we do, and that's an important perspective. The data that I'm showing here is a time series of chlorophyll A, and you can see from 2017 to 2019, color-coded by different lake, the blooms in Deal Lake in 17, 18, and 19, and then the further blooms in places like Capaccon and Sunset Lake in 2019. I talked about nitrogen and phosphorus earlier. If you go next, please. This scatter plot shows from the DEP data total nitrogen and total phosphorus plotted against each other in a correlation plot, color-coded by water body again. And what's interesting here is if you look closely at the colorings, the lakes tend to group out among themselves. And this tells us that, within New Jersey even, we've got our own trophic state gradient from lower to higher nutrient levels. And if you go next, please. If you look at nitrogen, and go next one more time, please. if you look at nitrogen and phosphorus plotted against chlorophyll A levels in these lakes, again, you can see the groupings by lake from lower nitrogen and phosphorus to higher, but they also tend to trend up and to the right with chlorophyll A, meaning that, now these are correlation plots, not causation plots, but this does give us a perspective that's important when we're looking at New Jersey as a whole, not just one lake at a time, as to the factors that are responsible for elevated chlorophyll A and also possible harmful algal bloom formation. And there's a lot more data that we're dealing with. This is some simple graphs from the first of data sets. There's cell and toxin data, there's rainfall data, and we're going to be working to put that all together and generate science-based answers to questions that have been given to us. Next, please. So I want to end and lead into Dr. Cortman's talk with this idea, which is not my own, and New York State has a similar strategy to this for a diverse strategy for mitigating and managing HABs. It starts with the big long-term stuff, dealing with climate change and reversing the direction that we're heading in on that front. Now, this is multi-decadal work and national to global scale work. It's not going to happen overnight, but it has to be kept moving forward. Next. On a more regional to local scale, there's watershed nutrient management, some of which we're going to hear about later today when Dr. Abrupta talks. This is also not small scale stuff and not short term stuff, but it also needs to be addressed and has to move forward. Next. And then in the meantime, while in-lake treatments and monitoring and forecasting are not going to solve the HAB problem, they're incredibly important tools in our toolbox that afford us resiliency to continue moving forward and using the environments as we take care of these bigger problems. And so as a strategy moving forward, I think it's very important that we move all of these measures together strategically to holistically address the HAB problem in New Jersey and the country. And on that, I'll end. Thank you for your attention.
Thank you, Jason. Next up, we have Dr. Bob Cortman, who is an applied limnologist, as well as the founder and president of Ecosystem Consulting Service, and he is another one of our expert team leads.
Thank you. Can everybody hear me all right? Can you hear me now?
Yes, we can hear you. Go ahead.
Okay, thank you. You've heard quite a bit about cyanobacteria, what they are, and our changing climate. Especially relative to climate change is increased variability, especially our winters, very shortened ice cover duration, much warmer than typical. You might want to make sure that that's not on automatic time flip. And some of the phytoplankton, especially the cyanobacteria, as Jennifer had talked about, are very resilient, very adaptable. Don't think of the cyanobacteria as being primitive. Think of them as being well adapted and able to outcompete many of the other phytoplankton. Next slide, please. Just for clarification of the terms that I use, the phytoplankton are all of those organisms that live by photosynthesis that are adrift in the water. And that includes the eukaryotic algae, the diatoms, the green algae, chrysophytes, and others, as well as the more bacterial types of organisms, the cyanobacteria. Within them, you have many different eco-strategists. You have some that become very buoyant. You have some that live in deep layers. Generally speaking, the eukaryotic algae are more or less desirable than the cyanobacteria. Next, please. So to manage and do something about cyanobacteria, the First step is to decrease the amount of available nutrients in the water body. And since the 1987 amendments to the Clean Water Act, much of the focus has been on watershed loading. And that's appropriate because the lake is a reflection of what comes from the land. So we need to reduce the amount of nutrients and sediments and other contaminants that get into our water bodies. We also need to deal with the processes that occur within the lake. Sediment release of nutrients and vertical transport are very stimulating factors for especially cyanobacteria. Next, please. Although everybody thinks of loading as being the dominant characteristic controlling phytoplankton in a lake, the other side of that trophic state coin is the removal mechanisms, the settling, grazing by zooplankton, retention, and outflow from the lake. So we need to manage both the loading and the removal mechanisms in the lake. Next. Some of the team members have a great deal of expertise with specific cyanobacteria genera. The life cycle. This is a life cycle of some of the more troublesome cyanobacteria, Aphanizomenon, Anabina, the ones that form those overwintering spores. And if we look at the life cycle, we may be able to identify particular points within that cycle where management is appropriate. Next, please. Phosphorus availability basically determines the amount of primary productivity that can take place in lake water, or how much. But it's all of these other factors, the light intensity, the ability to become buoyant, the grazing rate, the availability of silica for diatoms, and inorganic nitrogen for non-nitrogen fixers. All of these features determine what organism performs that primary productivity. Next, please. The team is also addressing specific questions posed by the DEP. Questions like, are there adverse effects of managing a lake with algicide, copper, routinely? And I don't think any of the team members would recommend copper sulfate as a routine management strategy. It is a tool that in certain circumstances can be an appropriate tool. So the team is going to be addressing these specific questions. Next, please. We're going to be looking at each of the potential management strategies, where they tend to fit most, what the risk of unanticipated adverse effects might be, cost effectiveness, advantages, disadvantages, but above all, the first lake management principle is whatever you do, do no harm. Next, please. Again, there are specific questions relating to that first principle, do no harm, and effectiveness. So the team will be looking at all the questions posed by the DEP. Next, please. So let's take a look at a suite of methods that can be used to do something about preventing, reducing the risk of harmful algal blooms, and perhaps even managing blooms when they do occur. These are methods for reducing loading to a water body. There are methods for watershed management that apply throughout the watershed. Generally, you want to get as close to the source of mobilization of nutrients and sediment as you can. Generally, you'll want to manage especially the first wash, the first flush of a storm episode. Next, please. And there are also management methods for wastewater for agricultural uses, and the team will be examining all of the various watershed management approaches for all of the different sources of nutrients. Next, please. There are also watershed management approaches that can be applied within this tributary conveyance system. First, flush capture, infiltration, creation of wetland renovation systems. Next, please. And there are also management methods that can make a lake able to deal with the loading that it receives without expressing the primary productivity as harmful cyanobacteria blooms. There are methods to remove nutrients from the water column, nutrient inactivation. There are methods to manage how a lake stratifies or whether the lake stratifies. And managing stratification is going to become very important because it's a way to adapt our management to our changing climate. There are aeration and oxygenation methods, sediment removal, mainly as a deepening tool, a sediment accumulation approach, sediment oxidation, and there are emerging technologies. Next, please. And of course, there are also methods to manage when blooms do occur. Algicide treatments, the newer non-copper algicides that treat by oxidation rather than toxicity show a great deal of promise, especially if we can identify those points within a life cycle of a bloom when the treatment can be really focused on preventing development. Can you go back? Thank you. There are also methods that are applicable to reservoirs that tend not to be as applicable to natural lakes. Reservoirs have a large water budget generally and have infrastructure that can be used for management. And with that, I'll conclude. Thank you.
Carrie, are you there?
Okay, thank you. We're taking a quick break and then we'll come back for some questions. I think there was just one question we had that's been up there for a long time, and that's from Paul Zarilla. Would you like to just ask your question? Paul, are you still there? Okay. We have a number of questions in the chat. Oh, Paul, go ahead.
I'm sorry I was late. I was having trouble getting onto the call here. But I just had a question. I don't know if this was covered or not, whether Greenwood Lake will be getting any monitoring buoy this season, and or what other help might be received from the DEP regarding HABs.
Vic, do you want to take whether or not there's going to be a buoy there?
Sure. So we're, like I said, we're getting 10 additional buoys. We're evaluating where they should be placed, based on priority, whether they're recreational, drinking water sources, and we're going to confirm where we're going to place the buoys with the expert team. So it hasn't been determined yet, but but more than likely, water bodies that have continuing issues with HABs will have a buoy placed at the water body.
Okay. When might we find that out, or you'll find out, if it's going to be one in Greenwood Lake?
We literally are just unpacking the buoys. We just received them last week. So we anticipate having them deployed by mid-May, so sometime before then.
Great. Okay, thank you. My other question was the report that the team is going to be putting together. Any idea when that is due? Because I think one or two of the part of the study is whether drawdowns are beneficial or harmful to HABs, and we are scheduled to have our drawdown this season.
So the report itself will probably, it's unlikely to come out prior to this HAB season. It will depend on the amount of data that the HAB team goes through and other factors, but we're anticipating to have a report out hopefully in the next 7 months or so, give or take, but it will probably be near the end of this, near this fall or after this HAB season, quote unquote season.
Okay, thank you. So we'll just proceed with our drawdown, and if we're asked to stop, we'll just stop.
Okay, thank you, Paul.
All right.
Ray, you had a question on time range of considering optimum flushing rates, considering one of the factors to be nutrient loading. I don't know if that's to Jason or Bob. Someone. So I'm not hearing a response to the question. So
this is Jason. I don't quite understand the question. And is the time range for optimum flushing rates? I don't know if Raymond
Ray,
is.
Yeah, Ray, do you want to expand on your question?
Yeah, Dr. Cortman, I believe you mentioned about basically hydraulic detention time in the water bodies. Obviously, it probably varies for different water bodies depending on size, nutrient inputs, nutrients and internal and external. You talked about detention, and is there a range that you might be looking at about flushing rates? What's more advantageous? A quick flushing rate or prolonged? Is there a range that you might be looking at, or is it so variable that you really can't go at the time?
Yeah, it's very ecosystem specific. Can you hear me?
Yes.
Okay.
Yes, we can.
It's very specific to an ecosystem. Overall, the more rapid flushing rate will tend to be better as far as avoiding cyanobacteria accumulation. Also, depth selectivity can be useful, especially in reservoirs. Reservoirs tend to have infrastructure where you have various release or supply intake depths, and you have a large water budget relative to the volume of the reservoir. So you can manage the water quality, especially internal loading and vertical transport, by manipulating where the water comes from that leads the lake vertically in the water column. So it really depends on the specific system. But in general, a more rapid flushing rate of the water body, it tends to be a healthy feature.
And in terms of that, it would probably be seasonality also. I mean, like early spring when everybody's fertilizing their lawns, and then you get a a rainstorm event, it might, water bodies might be full, then you get a flushing, but then you have a flushing of those nutrients also from fertilizer applications. So it's a double-edged sword there, I would think.
Correct. It's the seasonality to it as well as the annual flushing rate of the lake.
So monitoring is key.
Monitoring is key, and keeping track of those storm events and the changing climate. I've started using growing degree days like they use in agriculture to be able to anticipate what's coming during the summer. This year, for example, we've had a little bit more normal of a winter, albeit coming late. I mean, through December was incredibly warm, and then we got into some real winter weather in February. So, tracking those kinds of events can at least give you some indication as to what's coming.
Bob, thank you very much, and thank you for your questions. There are still a number of them in the chat. If we don't get to them one by one, we will answer them in the chat for you to the best of our ability, and we are keeping track of our questions, but in the interest of time, we need to move on. So at this time, I'd like to introduce Dr. Wang Zhang, who is the Associate Professor from Department of Civil Environmental Engineering at NJDEP, and he is going to be talking to us about a technique to remove HABs. Go ahead, Doctor.
Hello, everyone. Can you hear me well?
Yes, we can hear you.
Okay, perfect. Yes, so my name is Wang Zhang. I will be the PI for this HAB project, and I'm very happy to take this opportunity to introduce to you our project scope and project goals and plans. And as you can see that our project has the title of mechanical removal of HABs in lakes using air micro nanobubbles from a specialized floating platform. So this project aims to remove the algae using a broad array of different mechanical processes, such as nanobubble aeration, air flotation, membrane filtrations, with the very minimum use of chemicals, whatever chemicals, into the lakes or treated water. So in this project, we're going to team up with Meadowlands Environmental Research Institute and also an industry partner, Brightsea Group Incorporation. And we're going to collaborate on the project tasks that I will elaborate later. Next slide, please. Next, please. Next slide. Hello?
Are the slides not advancing?
Okay. Okay.
Here. Can you see that?
Nope.
Can anybody else see the slides?
Yes, I can see it, Jess. They were advancing.
Oh, okay. So okay. So now it's just updated. Okay. So this presentation will consist of the following agenda that I will basically start with our project team and team introductions. You can proceed. Next slide. So I have a little bit delay, so I'm not seeing the slides, but I'm going to just keep talking. I hope that everybody can see the updated slide. That will be the third
the relevant background slide is up.
All right, so it's just a couple seconds delay here. So since probably 2012, we've been working on the algae removal, or we call it algal biomass recovery, using technologies ranging from magnetic separations, as you can see from the picture on the bottom left. We use the nanoparticles to remove the algae from water, and using magnetic fields to enhance the separation. And then we work on the electrochemical membrane filtration, starting from 2016 under the funding support from National Science Foundation. And we're looking at the recovery efficiency using the membranes, and then devise a viable technologies to protect the water quality, but also recover some of the the nutrients and the material product that we can utilize. And then since the 2018s, we step into the nanobubble business, and we try to develop different kinds of nanobubbles, such as oxygen nanobubbles or ozone nanobubbles, to mitigate algae, especially harmful algae. And we're earning the support from EPA to to deploy this technology in the natural, or basically in the laboratory conditions. Okay, next slide, please. All right, so this slide summarizes the key, some of the selected publications we've published in the past few years, and we also launched a couple of symposiums, recognizing the importance of algae or algal recovery, algal removal. And next slide, please. And now we're going to just briefly introduce our partners a little bit. Mary, as you can see, is a part of the New Jersey Sports Exposition Authority, NJDEP, right now, but it used to be a part of Rutgers University before 2020. And Mary's has been dedicated to diverse long-term environmental monitoring project. As you can see, they cover the monitoring work ranging from water, air, and marsh surface elevation, all kinds of environmental media. Next slide, please. And Mary's lab is a NJDEP certified labs with very strong, instrumentation and the capabilities for a different number of environmental measurements, and they provide very complementary instrumentation power to this team. And for example, they particularly will cover the GIS survey and mapping for this project. Next slide, please. So in 2020, in September, even though in the worst time of pandemic, and Mary kindly offers some support and help us obtain some preliminary data. They flew the drones to capture the high resolution images and for the lakes in Branch Brook Lake. And then we also acquired the bathymetry data that allow us to understand the distribution of the lake depth. And that is very crucial for us to launch the boat. So in order to avoid the collision with the sediments or the lake bottom. And we'll also cover, measure some of the basic water qualities that is complementary to what we do at NJIT. So next slide, please. So Bryce is a consulting firm. It's an environmental engineering consulting firm localized in New Jersey. And we've been partnering on quite a few projects. And more importantly, Bryce is a member of ITRC. For those who are not familiar with that, it's Interstate Technology Regulatory Council. And particularly, we're going to team up with Bryce's technology concept validation and prototype development team. And that team consists of researchers like me and industrial designers, mechanical and electrical engineering, software developers, and prototype experts, which is provide very complementary skill set that we need for this project. Next slide, please. So particularly Bryce will playing a very important role in coordinating with the vendors and manufacturers and builders, transportations and all kinds of, key chains, for this project. For example, to build a boat, a highly functional boat like the picture I show you at the bottom left, it will involve the use of multiple kinds of equipment and instruments, and they're going to help us, reach out to those companies and vendors and help us fabricate this boat. Next slide, please. So last year also, again, we had a couple of interactions and meetings, just sort of having a planning and a preparation to get prepared for the work for this year. And due to the delay of our contract with the DEP, we just had a lot of meetings, and also we had some site inspections, visit at the Dew Lake and also Branch Brook Lake, and met the lake management teams and get their feedbacks and suggestions. Now we are ready to go. And next slide, please. So a little bit backgrounds on the project. So the rationale behind this, the choices of the two lakes, as you can see that the Branch Brook Park Lake and the Dew Lake will be selected as our targets for the demonstration research. The Branch Brook Park Lake is very close to NJIT in Newark, New Jersey, and in Essex County. And the Dew Lake is in Asbury Park and in Monmouth County, a little bit far away from NJIT. So these two lakes are selected because they were listed on NJDEP's website that were reported to be impaired by HAB in the past, and also they are relatively small and safe to launch the boat and operate without the concerns on the complicated hydraulic conditions, and it's very accessible to the the personnel who can who launch the boat, and so also they are very geographically convenient for our team to work with. So our expected project goals include we're going to develop a multifunctional air flotational platform, including other instruments as just mentioned. We're going to incorporate the membranes, filtrations, depending on the project progresses. And by injecting the air flotation, the micro bubble or nano bubble aeration, we're hoping to increase dissolved oxygen and sustain a high level of dissolved oxygen, and that boosts up the biodegradation activity, and that would essentially increase the degradation of water pollutants in general and reduce hypoxia and other factors that sort of trigger the growth of a HAB. And then we're going to continuously assess the water quality parameters, especially before and after the operations of the boat. And we'll demonstrate this sustainable mechanical platform to rapidly remove HABs from the impaired water, and we'll deliver a very comprehensive and smart bloom control strategy and suggestions to the community. And based upon the skills and experiences and results we're going to achieve down the road. Next slide, please. So here, I'd like to just briefly talk about the technology features. You may wonder, so what kind of platform we're talking about. As you see here, those are the pictures for a mid-scale and upscale or large-scale platform. You can certainly shrink the size or increase the size to incorporate different functions or increase the capacity of a treatment. So I just want to say that the technology here we present is different from other technologies or processes. We're not going to use a lot of chemicals. We hope to just physically take out the algae and reduce the regrowth of the algae by eliminating the nutrients there in water. So if let's say you kill the algae, but still there's the dead body still in the water, so the nutrients will be released to the lakes, and then they're going to foster the growth of the algae later on when temperature or other weather conditions are favorable to them. So we're going to remove and we're pretty close to a chemical-free process, even though we may add a little bit, benign chemicals. They're definitely not detrimental or leaving any adverse impacts on the ecosystems. And we can tailor the instruments and increase or upscale the instruments to have different capacities. As you can see, the boat could be shrink to you may proceed to the next image at that next slide, please. So this slide shows you a pretty small compact boat that we can fit in multiple instruments. As you see here, we can have first we have air pump and aeration system, water pumps and diffusers to diffuse out those micro bubbles, and then we have a centrifugation to dewater the algae that we harvested from the surface, and then we can further condition the algae biomass using membrane filtration. Depending on it's not to say that we're going to have that. It depends on the performances and the need for that, the dewatering requirements for the algae biomass. And again, we're going to separate the biomass, and then we'll properly treat them. Maybe just dispose them in the landfill because they are potentially hazardous material that releases cyanotoxin. Next slide, please.
Doctor, you have about 5 more minutes.
Okay, yeah, almost done. And so you can see that we had some preliminary work last year, and we had a lot of instruments calibrated and just get prepared for the environmental monitoring, especially we say that we want to monitor the treatment performances. We also structured the QAPP and communicated with the NJDEP to get this approved. And then, so next slide. So what plans? So this year, we already actually had conversations with our builders, manufacturers for the boat, and we hope to deliver the boat no later than June this year. So we are able to test run and get some, our drivers on the boat and train the team crew members. And we're all going to have a pretty, early stage testing and on a variety of tasks, that we propose to NJDEP. And in the winter time, we're not going to launch the boat because there are going to be no significant level of HAB. So we're going to summarize, compile the data, and report to NJDEP, and also share the work with the lake management team. And next year, we're going to launch the boat one more time to get, to validate and confirm the results. Next slide, please. So regarding the uncertainties and difficulties, this is something that we anticipate or something that we already realized. For example, the material cost and fabrication maintenance, it could be something that would is like a variable that we should keep in mind. It depends on the the treatment requirements, how large the lakes or the water body is. And then also the equipment, the pretty heavy, large equipment that would transport this here and there. It involves the use of a special transportation tools or something. And more importantly, we're going to train our personnel, driver, the boat drivers, and give them, skill set, the operation skills, the testing performances or the treatment performance might be affected by a lot of factors. For example, the water quality variations, weather conditions, and hydrodynamics. For example, the algae biomass are floating, are swimming. They're not dead body. They're always move around, and that could somehow interfere our treatment performance. But this is something we anticipate. And also your suggestions, feedbacks are very welcome and highly appreciated. And with that, I think the next slide will be, we'd like to thank our funding agency from NJDEP and also the crew members from the Branch Brook Lakes, Maya Loro, and the Don Braco from Dew Lake for their support and understanding. And thank you so much for your time.
Thank you, Dr. Zhang. Just reminding everybody that to please put any questions you have in the chat. We'll do our best to get to all of them after this session is concluded. With that, I'd like to introduce Dr. Fred Lubnow. Fred is the director of the aquatic programs at Princeton Hydro and Exton, and he will be speaking today about management activities to prevent and mitigate HABs in Lake Capaccon. Go ahead, Dr. Lubnow.
All right, thank you very much. Next slide, please. So just a little bit on Lake Capaccon. It is the largest lake in New Jersey. It straddles Sussex and Morris County, and about 500,000 people visit the lake or live in the watershed. There are 5 municipalities within the watershed. 4 have lakefront property. Next slide, please. So, and as a number of speakers have mentioned, there are 3 factors that will really trigger cyanobacteria HAB events: high seasonal temperatures, still water, thermal stratification, and elevated phosphorus concentrations. Next slide, please. And we've actually seen the impacts of climate change at Lake Capaccon. This is just taking the surface water temperatures for July for the last 30 years at Station 2, which is the mid-lake station, and graphing it. And we have a statistically significant relationship of an increasing temperature over the decades during the summer season. Next slide, please. So layered on top of that, in 2019, we got into this weather pattern in June where we would have these alternating short but intense storm events followed by a few days of sunny weather. So this is really what helped to trigger the blooms, not only at Lake Capaccon, but we saw similar blooms throughout New Jersey, New York, and Pennsylvania in a lot of the larger lakes that we manage. Next slide, please. And so these short, intense storms were essentially rinsing the watershed, putting in the nutrients, but not generating enormous loads of water that would help to flush the nutrients out. So it was enough water to transport the nutrients, and then we would have sunny conditions, and this was the result, these lake-wide blooms at Lake Capaccon. And HAB events and cyanobacteria blooms have been identified in Lake Capaccon, but they tended to occur prior to 2019 in September and October, essentially after the Labor Day weekend. So there wasn't that concern with direct contact with a lot of people at beaches. So what was unusual about 2019 is the blooms hit early in the season at the beginning of that high recreational season at Lake Capaccon. Next slide, please. And we just, in 2019, we just happened to be doing our routine monitoring. We were out there about a week before the bloom hit. So if you look at this graph, if you look at the last 2 points on the right, you have 2020 and you have 2019. You can see that each point represents an average concentration of the surface waters at Lake Capaccon, and each mean is about 11 measurements. So that 2019 phosphorus concentration was the first time in over 20 years we saw the June concentration exceed 0.04 milligrams per liter. And with Lake Capaccon's TMDL, we want to keep the concentrations at or below 0.03 milligrams per liter. So again, we had this warm, sunny conditions. We had these short-term storms hitting the lake, elevating the phosphorus concentrations above 0.04, and that's when we saw these blooms hit. Next slide, please. And this was the result. Beach closures, unpleasant conditions. Next slide, please. Now, so in response to the HABs, not only at Lake Capaccon but throughout the state, New Jersey DEP made grants available for the management, control, or mitigation of HABs. The Lake Capaccon Commission partnered with the Lake Capaccon Foundation, as well as Sussex and Morris counties, the 4 municipalities, Rutgers University, Princeton Hydro. We pulled together and submitted an application to implement a variety of in-lake or near or small-scale watershed activities that could help to control these HABs. On top of that, we were just completing an update of the watershed implementation plan for the lake, as well as Lake Musconetcon, and that funding came through the New Jersey Highlands Council. Next slide, please. So I'm not going to go through this slide because I'd rather spend the time going through the projects. So I'll go through project to project. Next slide, please. So the first project we implemented is we wanted to do a heavy treatment of Phoslock, which is a clay-based product that inactivates phosphorus, similar to alum, but it does not have aluminum. Instead, it uses lanthanum for the inactivation. It can be used to strip the water column of phosphorus, or it can be used to inactivate deep water phosphorus. We were using it in shallow waters in the southern end of Lake Capaccon to inactivate not only inorganic but organic forms of phosphorus. So that's one of the advantages of the product, is that it can inactivate both organic and inorganic phosphorus. It does not have the aluminum, but it is more expensive than alum or other similar products. Next slide, please. So we decided to focus on an area of the lake that has, it's relatively shallow, between 6 to 10 feet, a lot of organic material. So we wanted to inactivate that phosphorus. So we focused on the landing channel, southern end of Lake Capaccon. Next slide, please. It was a 50-acre treatment. It was one of the largest treatments of Phoslock conducted in North America at the time. We applied 22,000 pounds of the product at a rate of about 440 pounds per acre. So again, the focus here was to inactivate a lot of the phosphorus that's being liberated from the sediments. Next slide, please. And I'm not going to go into too much detail here. I just wanted to identify that we did have specific water quality goals for total phosphorus, for chlorophyll A, as well as Secchi depth. Next slide, please. And I don't have time to get into a lot of the data. I'm just going to show specific points. For example, what we did see was, if you look at on the further left, that 12th of June was before the treatment was conducted. The 24th of June was after the treatment was conducted, and we did see this this reduction in the soluble reactive phosphorus. We did see an increase in July of total phosphorus and total dissolved phosphorus, but that was after a fairly large storm event, and we saw elevated phosphorus concentrations throughout the lake. So that was more of an impact relative to external loading, but we did see SRP concentrations stay low, and we're going to continue monitoring this site well into this year to see how effective this treatment is over the long term. Next slide, please. We did see Secchi depth stay at or higher than our targeted goal. We wanted at least a meter of clarity, and we had that even after that storm event. Secchi depth was at a meter, and then it slightly increased going into September. Next slide, please. We also tried an alternative to copper, Green Clean. So it's a strong oxidizer. Next slide, please. So we did this at a beach in the township of Jefferson. And note that the beach it's on the main body of the lake, so it's not a cove or sequestered area. We did see an increase in clarity again with the treatment. However, we did see an increase in chlorophyll A and in phycocyanin, as well as the cell count. So even though it did clear up, it seemed like we did have more of the algae growth, but we attribute that to where that beach is on the main body of the lake. And this is something to keep in mind with any sort of management technique. Where are you relative to the hydrologic activities within that water body? Next slide, please. I'm going to talk real briefly about biochar. It's essentially burnt wood material. This is stuff that has this is material that has a high affinity for nutrient removal. We had some training sessions with the 4 municipalities, with the Public Works Department, and it was very well received for a few reasons. Number one, this material is relatively low in cost. And number two, once the material is used up, you can use it as landscaping mulch. Next slide, please. So we did some projects where we installed the biochar into these sleeves, into some stormwater ponds, into some streams, as well as in some existing stormwater manufacture treatment devices. Next slide, please. And just to go real quickly over some of the results, we saw a moderate to low amount of phosphorus removal in the streams. So 8 days after the biochar was installed, we were getting 55 to 58%, depending on the species of phosphorus. But after 3.5 months, we were barely getting any removal. So we may have to, if we're going to install these in streams, we may have to remove them at a higher frequency than every 3 months. Next slide, please. In contrast, in the stormwater ponds, after 3 months, we were still getting some pretty high removal rates. So you can see for total phosphorus between 67 and 81%. For SRP, we're getting 76 to 97% removal. And not surprising, what the manufacturer said is when you can extend the contact time between the biochar and the water, you get a higher removal rate. So we're playing around with this with different sized particles of biochar for the stormwater sampling, and we're going to continue to do some monitoring in this year, 2021. Those biochars that were installed in the streams, we're actually going to try to install them right where the streams enter the lake to see if we can increase that contact time. Next slide, please. So I'm showing this. This is Ashley Cove. This is a 2-acre area that's sort of isolated from the main body of Lake Capaccon. This is showing photos before and after we installed biochar. Next slide, please. But we also, at Ashley Cove, we were using Phoslock, but instead of a heavy dose, we were using very low dose treatments to try to reduce the phosphorus in the water column. And we did one green clean treatment to knock down the algae biomass. The one thing I want to point out is at Ashley Cove, tended to have some high phosphorus concentrations. You can see in 2012 in July, we were having an average concentration of 0.07. If you look at the figure in the lower right, you can see that total phosphorus, the highest it got through the summer season, with all those actions at Ashley Cove, we were getting at 0.04. So we did have a nice decline in the phosphorus. We're trying to get that down to 0.03, and that's what we're going to continue into this year, not only using the Phoslock and the biochar, but also installing some floating wetland islands. Next slide, please. Just wrapping up here, we are trying 3 different innovative aeration systems. We've installed 2 of the 3 in November of last year, so we'll be evaluating these nearshore aeration systems this year. One is an air curtain to disrupt still water conditions in front of a beach. The other systems are a nano bubble system with ozone, and then the third is just a nano bubble system. Next slide, please. So this is just showing the installation of the air curtain in November. Next slide, please. This is the nano bubble system with ozone, and then in about a month or so, we'll be putting in the third system, which is just nano bubbles, and we'll be evaluating all 3 this growing season. Next slide, please. So just to wrap up, we'll be cleaning out 2 large stormwater manufactured treatment devices and installing biochar to see what sort of effectiveness, phosphorus removal effectiveness we can get with that. And the Lake Capaccon Commission's been working closely with Rutgers on a very successful rain garden workshop program. That's been part of this program as well. And we'll be evaluating those nearshore aeration systems. Next slide, please. And I want to just, if we can just go to the next slide after this. I just want to show that, so last year we, on about 7 or 8 lakes, we used the phycocyanin meter as a surrogate, and we were collecting cyanobacteria cell counts at the same time. At Lake Capaccon, we got a strongly statistically significant relationship between the phycocyanin meter and the cell counts, which is really nice because we can now use the meter to sort of predict what those cell counts would be. So some lakes, like Capaccon, we got a nice tight relationship. Other lakes, we did not. So it's very lake specific. And I can say that what we've been seeing too is if you have a lake that's relatively shallow and has a lot of weed growth, plant growth, the relationship's not as tight. And it seems like flushing, the flushing rate of a system will really help to dictate whether or not the phycocyanin meter can be an effective means. But I would strongly recommend, if you're interested in this, that you sorry about that, that you strongly consider the use of cyanobacteria phycocyanin meters. I'm going to conclude since my dog's barking. Thank you.
Thank you, Dr. Lugnow. We seem to have that problem a lot with our pups. It's great. Thank you for that talk. And we're going to move right along now to Ms. Heather Desko. Heather is the Senior Watershed Project Specialist at the New Jersey Water Supply Authority, and she's going to be talking about a project at Spruce Run. Heather?
Thanks so much, Carrie. So good afternoon. It's officially afternoon, and I know I'm the last one between you and lunch, so I will try to get through this. And thank you for inviting me to speak today about our HABs mitigation project at Spruce Run. Next slide. So just a quick outline here. While many of the HABs mitigation grant projects are for in-lake management efforts, ours is in a tributary near the inlet to the reservoir. So I'll cover a bit of the background of the Spruce Run HAB and the site selection of the Mohawkaway Creek. And while we haven't yet installed the biochar, I will provide you with some plan information about our monitoring efforts and a project timeline. Next slide. The New Jersey Water Supply Authority is a state-created water authority in but not of NJDEP. The authority is tasked with operating and maintaining infrastructure related to water supply, specifically Spruce Run Reservoir with the red circle, Round Valley and Manasquan reservoirs, and the 60-mile long Delaware and Raritan Canal. In the Raritan system, the authority provides raw water to water purveyors who treat the water. Between the two systems, the authority provides water for approximately 2 million central New Jersey residents. In addition to the operation and maintenance of infrastructure, the authority's watershed protections programs, who I work for, engages in source water protection, planning, and management. The watershed office is situated right on Spruce Run Reservoir, which is the bottom left picture there with the arrow, which is quite lovely until you realize that there's a HAB, and then you just pull in and see that lovely green water every day when you pull into the office. Next slide. Spruce Run Reservoir is located in Hunterdon County. It's the third largest reservoir in the state and is 11 billion gallons, with the inline reservoir with two major stream inputs, the Mohawkaway Creek from the west and the Spruce Run Creek from the north. The land surrounding the reservoir is owned by the state of New Jersey and managed by one of three entities: the authority, the New Jersey State Park Service and Spruce Run Recreation Area, or the New Jersey Division of Fish and Wildlife with the Clinton Wildlife Management Area. I've highlighted two other features here with circles: the swim beach right in the center, which is part of Spruce Run Recreation Area, and the outlet tower for our water supply purposes on the southeast. At this point, we don't have a lot of in-reservoir water quality characterization data, but we do know a lot more about the contributing watersheds. The Mohawkaway Creek has an NJDEP approved watershed restoration and protection plan, and the authority also received a 2020 DEP watershed grant to develop a watershed restoration and protection plan for the other three Huck 14 subwatersheds that drain to the Spruce Run Reservoir on the east side: the Spruce Run Creek, the Willoughby Brook, and the Rocky Run, and which includes the reservoir itself. Next, please. Spruce Run Reservoir has been increasingly plagued by cyanobacteria blooms in recent years, including the fall of 2018 and then the extensive bloom from June 2019 through the present. Tributaries in both of the major watersheds have also had blooms in recent years, in the Mohawkaway Creek at Manny's Pond in 2017 and the Crystal Springs Preserve on the Spruce Run in 2018. Due to the length and scope of the 2019 to present bloom, it is likely that both internal and external nutrient loads are providing a consistent nutrient source for the cyanobacteria. Next. The current, and as we call it, ongoing, never-ending Spruce Run HAB was first reported in June of 2019, and the HAB has had many visual presentations over the course of these past almost 2 years, including even winter presentations last February. The dominant cyanobacteria taxa varies even between sampling sites on the same sampling date. So we do see a lot of the same usual suspects: the Microcystis, Warrenachinia, Dolichospermum, Aphanizomenon, as the dominant taxa. And sampling does not always match up with the worst case day, but we have caught some pretty bad events, including this 5.5 million cells per milliliter in 2019. And in 2020, we did have a new highest microcystin concentration at 38 micrograms per liter. The reservoir remains under a watch status at a minimum because the HAB never stays away for very long. And we have had both the advisory and warning status levels at 2020 in multiple locations. We have also done some additional sampling with USGS that has determined microcystins are consistently detected at varying concentrations. And then we have used these spat samplers in that third picture there to capture ephemeral toxin events that have also captured anatoxin A. And there's been some qPCR sampling that has captured the microcystin and anatoxin A and saxitoxin producing cyanobacteria genes in Spruce Run Reservoir. Next slide. So Spruce Run has been monitored through the DEP fixed wing aircraft remote sensing program since 2019. And these flyovers have provided us with some really interesting information about bloom dynamics and the potential external nutrient loads. So these screenshots are from June and July of 2019, the same time frame that Fred was talking about with those really frequent rainstorms that were happening every few days, and then it got really sunny. So I've marked some key features here to note. The yellow arrow is the inflow from the Mohawkaway Creek watershed. And then the red circles are at the swim park, the swim beach, and the blue square is at the outlet tower. So if you look at those yellow arrows, you can almost track the bloom coming in from the Mohawkaway Creek. You can see the higher concentrations, the estimated cells per milliliter there. And you can see that's then pushed with the prevailing winds right into the swim beach. So we've consistently seen this higher signature on the flyovers on the Mohawkaway side of the reservoir, which is one of the reasons we made this our focus. Next slide. So we had this inclination that the Mohawkaway might have been the problem in providing the significant external nutrient loads. So we did some estimates of total phosphorus concentrations at the Mohawkaway Creek at Van Sickle Monitoring Station, which the USGS has a continuous stream gauge here It's just 0.3 miles upstream from the inlet of Spruce Run Reservoir, and the site is also part of the state's ambient surface water quality monitoring network. So we've had quarterly water quality data there for quite a number of years. So the top figure is just showing estimated total phosphorus concentrations, and that they are higher seasonally in the summer months, and that the total phosphorus concentrations are generally increasing over time. And the bottom figure there shows that estimated total phosphorus concentrations have increased more significantly during the late spring, summer, and early fall, and more dramatically at higher flows. So the absence of having in-reservoir data to support an in-reservoir mitigation technique, we began to explore our options for in-stream mitigation and focused on the Mohawkway Creek as our target. Next slide. So we applied for and did receive the HABs mitigation grant for the installation and monitoring of an innovative project, and we selected biochar in the Mohawkway Creek to capture some of the external nutrient load to the Spruce Run Reservoir. The project is generally, it's relatively low cost, and most of the budget is actually for design and for water quality monitoring. Next slide. So Fred did mention a little bit about biochar. So when we were looking for the in-stream mitigation ideas, we were intrigued by biochar as a low-cost technique with the potential for really high nutrient removals. This, burnt plant material is highly absorbent, grabbing nutrients. There's really great nutrient removal data, but there has not really been a lot of research done in in-stream mitigation, really only in small ponds, but really for nutrient removal for algal mitigation. So we were interested to try this out, and it has the potential to provide a scalable and low-cost option for in-stream and or stormwater applications to address nutrient loading that contribute to HABs in the Spruce Run watershed and throughout the state. Next slide. So when the biochar is loaded in these porous bags and secured in an in-stream application, the water flows through the bag and the biochar, and the nutrients are attracted to the biochar, but the solids are not attracted. But they will be physically filtered due to the jagged exterior of the biochar. There are 4 what we call sizes of the biochar from this company, which is called Biochar Now, which we're going to be getting for our application. The smaller the size, the more surface area to attract nutrients. However, that means the slower the flow through the biochar. Since this is an in-stream application, we're going to be going with the chip size to allow for faster flow through the biochar. And as Fred mentioned, the biochar must be replaced on a regular basis. But for this purpose, there's no pesticide applicator license required to maintain the biochar. So it's something that's a little bit more accessible than something that requires an applicator's license. Next slide. So while the site is a great location for capturing nutrients not too far from the reservoir's inlet, there are some other complications. So the site is owned by the Water Supply Authority, but it's managed by New Jersey Fish and Wildlife Service. The Mohawkway Creek is a C1 trout production stream, so our best installation window that we're looking at is between March and June. The project has qualified, we've got some permit issues here. We qualified for a flood hazard area permit by Rule 32. We had the project reviewed through the land management review process through DEP, and we have also submitted a permit application for freshwater wetlands GP16 for habitat creation enhancement. A couple other things to note. Since we've mentioned that the biochar does hold water in its pore spaces, we wanted to make sure that we weren't negatively impacting the stream by backing up that water too much. And because the water is flowing through and over the bags, we know we won't see the high level nutrients that we might have seen if it was in a slower flowing situation. And knowing that we're going to have a lot of water be passing over the system, we'll need to make sure that the biochar is changed pretty frequently. And we're looking at about a 3-month changeover time to ensure the highest removal rates. Next slide. So our consultant is Princeton Hydro on this as well, and they have prepared these designs based on the site survey. So the project covers approximately 225 linear feet of stream and uses about 280 linear feet of biochar socks. The dashed lines are socks that will have protective cages, and the solid lines are ones that won't have cages. And the socks will all be secured with duckbill anchors to the stream bed. There are several configurations and designs, generally focusing on increasing the contact time without backing up too much water and affecting stream flow. Several of the designs you might recognize as they commonly mimic stream restoration designs. Next slides. These are just a couple designs. This is not all of them, but you can see the top picture here. This is right at the Charlestown Road Bridge, and that's the USGS gauge station in the top corner there. The goal of the is to ensure that biochar logs are below the water level at base flow for maximum contact time. And so we have this half moon design in the top picture that's using the vegetation, the little sandbar there, for natural protection. And the bottom picture shows the logs are installed parallel to the shoreline that can provide some bank stabilization. Next slide. The top picture of this design shows the logs are lining the perimeter of two large pools here. And the bottom picture shows a check dam style design for a section of stream that has a uniform width and depth. Next slide. And just a quick schematic to show the socks. Some of the socks will be without protection, and then some of them will have protective cages, and they will both all be secured with duckbill type anchors. And we'll be able to get into those cages to switch out the biochar as we need to for the project. Next slide. And of course, no demonstration project would be complete without a comprehensive monitoring program. So our analysis is going to focus on identifying the upstream versus downstream differences in nutrients during base flow and storm flow conditions. We're going to be deploying 2 ISCO avalanche refrigerated sampler units to capture the base flow and storm flow samples upstream and downstream of the biochar installation. Base flow monitoring will occur twice a month during the growing season and once a month during the rest of the year. And then we hope to have at least 3 stormwater samples to be collected every season with the goal of hopefully getting small, intermediate, and large scale rain events if the weather cooperates. And we're going to be doing some discrete sampling there and looking mostly at the total nutrients rather than speciating that out. Next slide. So for the data analysis, the statistical differences will be evaluated using R. And some of the questions we're hoping to answer are: to what degree of pollution control under the base flow conditions does this provide? How does performance vary from pollutant to pollutant? And is there any variation in the performance between large and small storm events? Next slide. And when we applied for this grant, it was pre-pandemic, so we are a little behind schedule from what we had anticipated. So there are a lot of moving parts with this project due to stakeholder engagement, site constraints, need for permits, and developing the quality assurance project plan for monitoring. So we do anticipate that we will be able to install the biochar monitoring equipment this month. Or the monitoring equipment be installed this month. And we are just waiting on the permit approval so that we can get the biochar installed in stream. But hopefully we are looking at March through June of this year to be able to install that biochar and then get this project going. And then we hope that it will be installed for a minimum of 2 years. And if it goes well, it would be something that we could potentially continue because it is so low cost and easy to maintain moving forward as much as possible, and the partners agree to. So we have had, you can just see we've submitted our QOP to DEP and Office of Quality Assurance and have received those and are just finalizing those. But we are pretty close to getting this in the ground. And when I signed up to do this presentation, I had hoped that we would have be very close to be able to install this. Next slide. And again, thank you so much for having me here today.
Heather, thanks so much. So we are at questions. And here's what we're going to do. We have a number of questions. We're asking you to put them in the chat. I'll call on you and then you can unmute your mic and ask your question. For those questions that we don't get to today, we will provide you answers and we'll post them on our web and send you the link to those questions and answers along with the recorded session from today. So with that, the first question is from Larry Supp. He has actually 2 questions. You want to unmute your mic and ask your questions, Larry?
Yeah, I think Dr. Lubno answered me already in the chat. My questions were in the Capaccon, since they installed a sewer system, has there been any benefit seen in the phosphorus loading since then? And secondly, is there any research being done on trying to harness the idea of heat exchange between the cold part and the warm part of a stratified lake?
Yeah, and this is Fred Lubno, and so I can just elaborate on those two. So for the sewering, we have a 30-year database at Lake Capaccon, and we did see that in the section of Lake Capaccon, in the borough of Capaccon, where you have the River Styx Crescent Cove section of the lake, we did see a decline in phosphorus concentrations once sections of that watershed were sewered. So very frequently, phosphorus would get as high as 0.1 milligrams per liter in there. And I think the highest we've had since it was sewered was maybe 0.08 at the most. So we have seen a decline in phosphorus thanks to the sewering. And the commission has been implementing a wide variety of watershed techniques over the last 12 years. And, for the TMDL, we're only about a third where we need to be. So we still have a ways to go, which was why it was very, even before the HABs hit, the New Jersey Highlands Council provided funding for us to update their management plan since it was over 12 years old. And then on your second question, as part of a 319 grant, we are evaluating thermal stratification and the internal phosphorus loading, again, the 30-year database we have, to see the influence of weather and how it can affect thermal stratification as well as internal loading, and whether or not internal loading accounts for a larger portion of the phosphorus available to the cyanobacteria over either the summer or the growing season in general. When you look at it on an annual basis, internal loading is less than 10%, which is one of the reasons why we focus so much on the watershed at Lake Capaccon. But on a temporarily shorter basis, so during the summer or during the high summer season, which I always describe as between Memorial Day and Labor Day, is that percentage a lot higher, and is that driven primarily by weather? And if it is, climate change may actually be exacerbating that internal phosphorus load.
Can I just make a quick follow-up question? Is that an easy calculation or a hard calculation to figure out the internal loading?
Well, it's to really do it, you need to have some decent water quality data. At a minimum, you should have temperature and dissolved oxygen profiles from surface to bottom at the deepest section of the lake. You should have multiple readings throughout the growing season so you can get a feel for the extent of anoxia in the deep waters. Because what happens is when you run out of dissolved oxygen at the bottom waters, that bond between iron and phosphorus is broken. That phosphorus can leach up into the overlying waters. And during storm events, that phosphorus-rich water can be transported up to the surface, but also you have many of the cyanobacteria have gas vacuoles, so they can move up and down the water column, take advantage of some of that phosphorus-rich water right at that thermocline. So there's a number of interactions that occur, but at a minimum, you need data like that. It would also be nice to have data on the phosphorus concentrations, iron, some other parameters immediately above the sediments as well as the sediments themselves. But at a minimum, you should have some pretty good data on temperature and dissolved oxygen through the water column.
Thank you, Fred, and thank you, Larry. Our next question is from Steve Sousa. Steve, you want to unmute yourself and ask your question?
Yeah, thank you, Carrie. I'm not sure if it's a question for Dr. Lubnow or for DEP, but with respect to the use of ozone, because technically the ozone is being used to kill algae, is a pesticide permit required for that, or some type of special use permit required for that? Did you get that?
We got it. Any of our panelists or maybe someone from DEP, do we have an answer to that question?
Sorry, I didn't mean to stump DEP.
No, we're all on mute, so that's part of the problem. There's a delay response.
Carrie, this is Bruce. Can you hear me?
Yes, we can hear you.
Okay. Steve, I think we need to get back to you on that. I'm not comfortable just answering that right now. I think what we're going to do is some of the questions that we can't answer, we'll come back to and post with the materials from the summit. So we'll make sure that we note your question. We'll do a little research, make sure we got our answer right, and then we'll post it back at a later time.
Yeah, that would be very much appreciated because I have received numerous questions in the past about the use of ozone, in lakes and, state open waters. So thanks very much, Bruce.
Yeah, I mean, I would think that possibly, depending on how the installation is, you may need a land use permit similar to the biochar. But I would, need more detail and need to step back and do a little research.
Okay, thank you.
Yeah, and again, this is Fred. Just for my part, when we put in for the biochar and the ozone, we had a permit by rule specifically for Lake Capaccon, but I certainly did not want to speak for DEP when it comes to any sort of decision that they may make on ozone.
Fred, do you know which PBR that was?
Offhand, I don't know. I know Kelly's the one who put that together, but I can forward that to you, Steve.
All right, thank you.
Thank you. Our next question is from Robert McDowell. Robert, do you want to unmute yourself and ask your question? I'm sorry, it's Robert McDonald. I apologize. Do you want to unmute yourself?
Hello?
Yes, go ahead and ask your question.
Oh, okay. I just had a quick question. I have a lake in our senior citizen development, and it's hard to get the HOA to react to anything if you can't produce facts. So I was wondering if there was an economical way to test the water and confirm factually that you have a problem that they have to deal with. That's it.
Thank you. Anyone? Vic, perhaps?
I just, again, this is
sorry,
Fred.
I was unmuting
I'm sorry, go ahead.
Yeah, I responded in the chat. So the phycocyananometers, the field meters that Fred was talking about, they're a good way for to get a good approximation of cell count. They don't replace lab analysis. They can't be used to measure toxins, but they do give you a good idea if a HAB is present in considerable concentration. And I'm going to be speaking about that later in the summit. We're going to have a loan program for partners for these meters.
Thank you, Vic. Maria Petix-Kent, and I apologize if I mispronounce anyone's name. You're asking about how do you dispose of biochar once it's spent? Fred, yes. Do you have, or Heather, do you have an answer that you can provide?
This is Heather. So biochar can be used as a soil amendment afterwards. And I think even Fred has mentioned before that there are companies that will buy it back. I know Fred may have more information on that.
Yep. Yeah. They're like Heather mentioned, like biochar. Now they said that they'll actually buy it back and then they resell it as a soil amendment. However, for most of our lake clients who are interested in this, and again at Lake Capaccon, all 4 towns participated at a public meeting where we showed them the biochar, how you can create the sleeves, and then they were like, "Oh, well, this number 1, it's not expensive. Number 2." Show us how to do it. And number 3, if we can use it as landscaping mulch, that's fine with us. We'd rather do that. In the past, some of the other media we've used for some of the stormwater treatments, number 1, the media tended to be relatively expensive. And number 2, they were like, well, how do we dispose of this? So here where you're talking about something that you would replace maybe, 2 to 3 times a year, and then you're using as landscaping mulch, they just would rather just use it as mulch as opposed to try to set up some deal selling it.
Okay, all right. So it wouldn't be considered a waste of any sort. It's just take it out of the bags and put it on the put it anywhere along the shoreline.
Yeah, using it as landscaping mulch. I mean, I wouldn't use it. I mean, ag people I hear ag people will use the stuff as a soil amenity, but I would just say use it for landscaping. It's, obviously if it has those nutrients, bound up in that material, once you put it in as some type of soil amendment or mulch, you want those nutrients to be available for the terrestrial plants as opposed to the aquatic plants.
Okay, thank you. Thank you very much.
Okay, I know that there are more questions and I apologize, but we're going to have to stop here so we can take a break. We have a 30-minute lunch break. When you come back, we invite you to please use the same link that you used to get in this morning. We have a very full agenda this afternoon as well. We're going to be, there's a session on science, and we're going to be talking about best management practices, HABs, impacts to drinking water, and there'll be a discussion on finance options for HABs, as well as partnerships that we can forge with all of you. So we look forward to seeing you later this afternoon. Any questions that were unanswered, we will address them in the chat. And as I said previously, other questions that need further research, we'll do that research. We'll put together a link for our website and send that out to all of you. So thank you very much for your participation this morning, and to all of the speakers, very informative, and we look forward to joining you in about 30 minutes. Thank you.